My name is Jakob Bellmund, and I want to share some data on memory for sequences of events with you. And this is a neuroimaging project, and I hope to convince you that event memories in the hippocampus store actively constructed event times and that they generalize the temporal relations they represent across different sequences. And so the notion that memory is a constructive process is not new, right? So this probably goes back at least almost 100 years to Bartlett, for example, who pointed out that our memories are not veridical records of the past, but rather that remembering is a constructive process. And today I want to talk about constructive memory for time. And so in some circumstances, temporal memory can also be constructive. So consider, for example, the question, when did your mom call yesterday? Right? So the precise time when your phone rang might not be part of your memory, but you do remember that you were watching a football game and you know that those start at 9, at least if you're in Central Europe. And you also recall that you went to bed at around 11 and somewhere in between she called. And so based on these information, you estimate that the call took place at around 10 p.m. So what you're doing is use the temporal relations of events from the episodic sequence that is your memory of last night, together with associative, semantic, or contextual information to construct when this specific event happened. And that's what I mean when I say that memory for time can also be constructive. And in the example I just gave you, one of the things that you use to estimate the time is based on the sequence of events that unfolded last night. And these sort of event sequences are a key feature of episodic memory. And we know that the hippocampus and the adjacent entorhinal cortex are particularly relevant to processing sequences and forming these sequence memories. And we've attempted to summarize some of this literature in a recent review that was published in the Journal of Cognitive Neuroscience. And I want to highlight some of our own previous work to show that temporal relations of events are reflected in multivoxel patterns in the hippocampal-entorhinal region. So we ran an experiment where participants navigated along a fixed route through a virtual city, which is the blue line here, and they would encounter events at the positions marked by the blue circles. And we scanned participants after they learned this event sequence. And what we saw was that the hippocampus and lateral entorhinal cortex formed sequence memories that sort of mapped the structure of these sequences so that temporal relations between events were reflected in the multivoxel patterns. So what does that mean? So the similarity of activity patterns was correlated negatively with the temporal distances between pairs of events. And so to break down that correlation a bit, what this basically means is that events that have a low temporal distance between them or are close together in time, that these get relatively more similar than those events that are separated by larger temporal distances. So this gives rise to these negative correlations. But is this due to mnemonic construction, or is it simply because participants chain the events together and you have some sort of order mechanism? Or is it the actually elapsing time between events that underlies such an effect? And this is the first question that we wanted to address in the project that I'm going to present today. Do sequence memories in the hippocampus reflect mnemonically constructed temporal relations, or do they more tightly relate to the order of events or the elapsing time between events? But first, let's briefly come back to why we think that memory is constructive. And so one influential idea is that we use memory-based constructions to simulate possible future scenarios. And there are different versions of this or similar ideas. And one important feature is that these constructions typically use information that is stored in memory to construct the future. And in particular, we often generalize across individual past episodic events or sequences and use general abstracted semantic knowledge when constructing future scenarios, which is something that's particularly prominent in the semantic scaffolding hypothesis. And so we can use these constructions to guide adaptive future behavior and to generalize to novel situations. So if we generalize across experiences to anticipate the future using constructive memory, then one possibility is that we construct an integrated representation of multiple event sequences that actually generalizes across the different sequences. So here, this is one of the questions that we ask here. So to illustrate this, consider a second sequence with different events, in addition to the one that I already told you about. And now, if our mental representation of these sequences share an underlying structure, which could be our day-to-day rhythms or the reference frame provided by the circadian clock, we could in principle integrate them into one representation. And figuratively speaking, this is like organizing them along the same mental timeline when thinking about when these events occurred. So the temporal relations of both sequences could essentially be represented along the same underlying dimension. And so the second question I want to address today is whether hippocampal sequence memories generalize temporal relations across sequences. And to get at these questions, we ran an fMRI study consisting of 4 parts, and I'll walk you through these as we go. But I want to highlight that a lot of this work was done by Nicole Montaigne and Lorena Deuker, who was a postdoc in Christian Dell's group at the time, and actually collected these data together with Nicole. And I'll start with what I'll call the day learning task, which is at the heart of our paradigm. And so in this experiment, we asked participants to learn the times when different events of a sequence took place. And we told them that each of 4 sequences corresponded to a day in a family's life. And the events that we showed them were images that were generated using the computer game The Sims, so their everyday activities like reading a newspaper or cleaning the bathroom. And crucially, we asked participants to infer when these events took place relative to a hidden virtual clock that ran for each day. And this hidden virtual clock was revealed exactly once in between presentations of 2 events. And so, for the remainder of the task, the screen was black. The duration of these black screen periods scaled with the amount of time that was passing, of hidden clock time that was passing. And so, to solve the task, participants had to keep track of actually passing real time and combine it with these time cues to construct the specific time of a given event. So for example, the first time cue here is 8:28. And then the screen goes black, and at some point the reading the newspaper event occurs, the screen goes black again, and then the second time cue, 10:04, is shown. And so if you keep track of passing real time between these time cues and the event, you can use mnemonic construction to infer that the event was actually at around 9:30 a.m. And so importantly, the virtual time, so the hidden clock time of each event was fixed, but the current virtual time, so these time cues, were varied on each repetition of the sequence, so that participants could not simply use an associative strategy to solve the task. And of course, we wanted participants to learn these virtual times of events. But it could actually be that they only picked up on the order of these events, so 1, 2, 3, 4, 5, as each sequence had 5 events. Or it could be that it's the actual real elapsing time between events that underlies their learning. And to partly disentangle these different time metrics, we varied the speed of the hidden clock. So there were 2 sequences where the hidden clock was running fast and 2 sequences where the hidden clock was running slow. So here, the 4 sequences are plotted in real time. That's the x-axis here. And what I'd like you to appreciate is that for the slow clock days, virtual time passes from around 9 a.m. to 6 p.m. or 7 p.m. And for the fast clock days, it goes from around 7 a.m. to up to 10 p.m. So more virtual time passes in the same amount of real time. And so this manipulation partly disentangles these different time metrics. So of course, this is a memory experiment. So we asked participants whether they actually learned the structure of the virtual days. I'm going to focus on one memory test today, which is what I'll call the timeline task. And in this task, participants saw the 5 events that would make up one sequence. And the task was to drag and drop these events onto the timeline to indicate when they thought each event would take place. And if we look at the raw data from their responses, then we can see that they are fairly accurate at doing so. So let's look at this plot. Each row corresponds to one of the sequences, with the gray line at the bottom denoting the true times of events with the circles. And then on top of each event, you see the distribution of raw participant responses with single responses, box plots, and distribution curves. And what I'd like you to take away from this is that the distributions of responses are nicely centered on the specific events. And indeed, if we quantify the errors that participants make, then on average, the absolute errors are just below 1 virtual hour. So participants form or very accurately construct and remember these event times. And now you can imagine that you can probably do okay in this task if you simply remember the order of events, right? And just evenly space your responses along the timeline. And so we wanted to know which time metric really, or whether the virtual time really underlies participants' responses. So what we did is we regressed the constructed time on the different time metrics using a summary statistics approach where we run a linear model for each participant, as well as a mixed model approach where we fit one model with all participant data and account for participant-specific effects using random effects. So, and what we see is that the virtual time of events explains the constructed event times, even when it's competing for variance with order in real time. So participants' responses are really driven by their mnemonic constructions. And with that, let's turn to the neuroimaging data. And the main question here is whether event representations in the hippocampus change to reflect the temporal structure of the different sequences. And to test how these sequences are represented in the brain, we scanned participants before and after learning the events or learning the sequences. And importantly, during these pre and post learning scans, we showed the events out of sequence. So we showed them in random order to cleanly estimate multivoxel patterns going back to each event. And so what we do is we use a technique called representational similarity analysis, where we aim to quantify changes of event representations. So what we do is we calculate the similarity between the representations elicited by pairs of events, which gives us, and we do this, which gives us a 20 by 20 matrix, which is reflecting the event similarities in both the pre-scan, so before learning, and in the post-scan, after learning. And what we do is we subtract this baseline similarity from the post-learning scan to isolate what I'll call representational change. So we want to isolate the part of the representation that changes through the learning experience. And now our question becomes whether these changes in representations can be explained by what participants learn in the day learning task. And in particular, we ask whether they can be explained or these changes in similarity between pairs of events can be explained by the temporal distances between events. And of course, we can quantify these temporal distances between events using different time metrics, right? So if we use virtual time, then these two events are 2 hours and 15 minutes apart in virtual time. They're 1 order position apart, or they're 9.3 seconds apart. And what you can appreciate is for different order positions, or for the same order positions, you can have different virtual times between events. For example, here in this first 2 examples. And importantly, we can do this for events that belong to the same sequence, which is what I'm showing you here, or we can do this for events that actually belong to different sequences to look at this potential generalization effect. And so we ask whether we can explain this representational change by the temporal distances between events. And we do this using a summary statistics approach where we fit a linear model for each participant and use permutation tests, and using a linear mixed effects approach where we are interested in the effect in fixed effects and can capture subject-specific effects using random intercepts and random slopes. And so in this quick overview, I'll focus on data from the anterior hippocampus. And we have a preprint out if you are curious about what's going on in the entorhinal cortex. So the first question is, when we look at pairs of events that belong to the same sequence, can we explain the changes in similarity with virtual temporal distances? And indeed, that seems to be the case. So we observe positive correlations in both the summary statistics and the mixed model approach between temporal distances and pattern similarity changes. And importantly, what this means, and this is somewhat surprising given our prior work, is that pattern similarity increases with virtual temporal distances. And I've broken that down for you here based on a median split, just showing you that for events that are separated by a low temporal distance, pattern similarity decreases, and for pairs of events that are separated by large temporal distances, representation of change is positive, so patterns get more similar. And next, as for the behavioral data, we wanted to ask whether this is really due to the virtual time of events, or whether it's actually real time or the order of events that explains this. And so, just like for the behavior, we ran multiple regression models to find that virtual time significantly predicts hippocampal representational change, even when it's competing for variance with the order of events or the real time that elapses between events. So these hippocampal sequence memories do seem to reflect the constructed times of events. And to get out our second question, where we wanted to look at the potential generalization of constructed event times, we tested whether the similarity of event representations changed according to virtual time, but now for events that belong to two different sequences, which is shown in red here. And if we look at the red bar here, then we can see that there's a significant negative effect. So we observe consistent negative correlation between temporal distances and pattern similarity change for events that belong to two different sequences. So this is exactly in line with the effects that we've observed before. So pattern similarity decreases with virtual temporal distances between events. And importantly, there's a significant difference between the effects for events that belong to the same sequence or to two different sequences. And so to kind of illustrate this, this difference a bit more, I'm showing you again the raw pattern similarity change values in blue for events from the same day and in red for events from different days. And as I tried to explain before, for same sequence events, similarity is high when events are separated by a large distance. The opposite is true for events from the same sequence, for events from different sequences. So across sequences, similarity is high when events happen at similar virtual times and is lower when events take place at more distant virtual times. So the way the hippocampus reflects these constructed event times depends on whether the events are from the same sequence or not. And to take this a bit further, I was wondering whether these representations of relations within one sequence or across one sequence that we see in our hippocampal RI analysis are the same, or whether they go back to different mechanisms, for example. I wanted to look whether these representations overlap anatomically. So I ran searchlight representational similarity analyses. And when you do this, focusing on the within sequence events, so pairs of events from the same sequence, then we see the same positive effect, which is significant in the bilateral anterior hippocampus. And what I did then is to ask whether in this cluster, so in this part of the brain where the same sequence effect is significant, can we also detect the different sequence effect, which is an independent contrast. So I defined a region of interest based on the same sequence searchlight and ran the across-sequence generalization analysis. And indeed, it was showed this across-sequence generalization effect in the cluster defined by the within sequence searchlight analysis. And if we run the generalization searchlight itself, then we can nicely see that it overlaps, or the results that it gives overlap with the same sequence effects. So I'm showing you in blue the same sequence effect, and in red the across-sequence generalization effect. And what you can see in the rightmost column is the overlap, which is plotted in yellow, which actually takes up most of the clusters. So these representations of within and across-sequence relations, they overlap in the anterior hippocampus. And so now we've seen that the hippocampus seems to hold event representations that carry information about the relations of events, even when they are from different sequences. And now in the remaining few minutes, I want to briefly return to the behavioral data to address the question whether this generalization across sequences could actually lead to biases in the event times that participants construct. And the idea for this analysis is inspired by reports of how past statistics affect sensory motor timing behavior, or how prior knowledge can bias our memories. And so what I did here is I wanted to ask whether for a given event, whether the times of the events at the same sequence position, but in the other sequences, whether they bias the remembered time of that specific event. Now that's a complex sentence, and I'll try to illustrate this. So consider this example where we want to look at the estimated time of the fourth event in the bottom sequence, right? So I'm zooming in on the general temporal structure, and we have 4 events that each occupy the fourth position in their respective sequence. And we're interested in the virtual time that participants construct for this event. And what I did now is I calculated the mean time of the other 3 events, and I asked, when is it relative to the time of the event of interest? And my question is whether this mean relative time of the other events at the same sequence position biases participants' estimate for this event time. So if we consider, or if we plot these data, then I'm going to show you on the x-axis the relative time of the other events, and on the y-axis the error that participants make in this timeline task. So values of 0 on the error dimension mean that participants are accurate, positive or higher values mean that they estimate a later time, lower values that they kind of undershoot in their time. And I'm showing you here example data from one participant who shows a medium strength effect, and each data point is one event. And what you can see is that there's a positive relationship between the relative time of the other events and the error that this participant makes. And if we look at our entire sample, so now each panel is the data from one participant, then you can appreciate that that this positive relationship of the relative time of other events and the timeline error is remarkably consistent across this sample. So when the other events happen relatively late, participants tend to overestimate or overshoot in their estimate for the particular event time. And of course, we can do group level statistics on this using summary statistics and a mixed model. And we observe that these constructed event times are indeed biased by knowledge about the event times in other sequences. And in our preprint, we describe a replication of this effect in an independent sample. And so this brings me to the end of my talk. And to briefly sum up, our data show that participants form fine-grained memories of the virtual day structure. I haven't shown you, but you can find it in the preprint. They remember which events form the different sequences, and they successfully construct the event times from the time cues and their experience of passing time. And it's really this constructed time that underlies their responses. And similarly, in the hippocampus, we detect that representational similarity scales with temporal distances between events of the same sequence. And again, this goes back to temporal distances relative to the virtual times of events. And intriguingly, temporal relations also shape representations of events that belong to different sequences. So knowledge about the temporal structure of the sequence is generalized. And anatomically, the within and across sequence effects overlap in the anterior hippocampus. And one slightly surprising thing that we found is that in the hippocampus, there's a dissociation between the way relations of events from the same sequence are represented compared to the generalization across sequence. So this is the positive correlation for same sequence events and the negative correlation for across sequence events. And what I haven't shown you here is that this is different from what is going on in the entorhinal cortex, which uses just one general representational format. And then in the last part, I showed you evidence that the constructions of the individual event times are biased by generalized event times from the other sequences. So structurally similar events, so events that have the same sequence position, but in other sequences, influence how specific event times are constructed from memory. And this is very much in line with work in other areas, showing how knowledge about general statistical properties can bias constructive and mnemonic processes. And it's further related to the semantic scaffolding hypothesis, which suggests that semantic or general knowledge provides a scaffold for episodic constructions. And so our data shows that knowledge that is abstracted from other events is used for our mnemonic construction of specific events. And with that, I'd like to close, and I'd like to thank you all for your attention, and don't hesitate with questions.
Thanks a lot. So we can take a couple of questions. Yes, Ben.
Hi, really interesting data. I'm struggling to understand, I think it's your key result, the dissociation in the hippocampus, showing that as you increase the temporal distance of events within a sequence, you get increase in representational similarity, versus you get the opposite across sequences. The opposite makes more sense, but why would representational similarity increase with temporal distance within a sequence? That doesn't make any sense to me.
Yeah, so I agree that this is surprising, and this is also not what we expected based on our prior work, right? Where the sort of negative correlation is actually what you expect, right? Because things that are close together become more similar. One speculation, so I can only speculate, I don't think I have the definite answer. So one speculation is that it's due to the increased associative memory demands that we're putting on people, right? So in the previous work, participants were learning only one sequence. And what happens here is that participants have to learn actually multiple sequences. And so you don't only have to learn the time of each event, but you also need to group those events together that belong to the same sequence. And one hypothesis is that this alters the way that similarity relates to temporal memory. There's actually some data from that can be interpreted in this way from papers by Laila Lavacci and Sharon Ranganathan, where they looked at order memory judgments and the way hippocampal pattern similarity diverges, where the presence of event boundaries actually altered how similarity and order memory were related. A second possible explanation, which explains maybe the low or the decrease in similarity for events that are close together in the same sequence, is the idea of hippocampal pattern separation or differentiation, where you push things apart that are very similar. In our case, these are things that are in the same sequence and nearby in time. But I don't, or this is an explanation that basically can explain this effect. It doesn't really account for the across sequence generalization effect, I think.
Okay, I'll have to think more about this, but interesting data.
I guess I'll ask a question. So I guess the effect of the virtual clock is to, if you think about temporal context signals, right, it may be to speed or to slow down the rate of evolution of temporal context in these different brain regions. So in light of that, could you maybe go a little bit more into maybe the differences between the entorhinal and the anterior hippocampus? Because we do know that we have these kinds of temporal context signals when you measure from neurons in the entorhinal cortex. But we also have the same kinds of signals in the hippocampus as well. So what makes these two regions do slightly different things in terms of one generalizing and one having the same representational format, you know, both within and across sequences?
Yeah, I think that's a great question. And so I think, all right, so maybe I should, can I unhide this? 1 second. So first, maybe the question, what does the entorhinal cortex do in this task? So in the entorhinal cortex, there's no difference between what happens in the same and in different sequences. but rather we see a consistent negative effect. The way that you would expect, that events that belong together or that are close together in time are more similar than those that are far apart. And so I think your question about temporal context is I think an important one, and I'd love to look more at what happens in the brain during the learning task, whether basically the rate of change is different depending on the clock speed, right? If you can actually speed up the way these patterns evolve. And I mean, so I think you alluded to data from the entorhinal cortex and hippocampus in terms of temporal coding neurons. So so in the entorhinal cortex, there's prominent work from the Moser group, for example, that shows that you have these linear decaying basically these traces of prior experience, and that you can decode time from population activity. And then in the hippocampus, you have time cells, famously described by Howard Eichenbaum, which basically fire at specific moments in time during repeated delays. And I mean, computational work sort of suggests that you can get time cells from these entorhinal inputs. And I think my speculation could be that it is the associative memory component of the hippocampus that sort of alters what happens then in the hippocampus, that gives rise to these differences between these two regions. But that's a speculation at this point, I have to say.
Cool. So yeah, it's 8:35 here. So thanks again to both of our speakers for very interesting talks, and thanks to all the participants. Yeah, so that's an applause sign, so not a question. So I get it finally. Well, thank you all for joining again, and take care. Bye-bye.
Thanks for coming. Thanks for your questions. Bye. Bye-bye.